Hi, my name is Ana Magos and this is my answer to question number 3. In this answer, I will be describing the physical methods of microbial growth that are effective at killing gram-positive bacteria and endospores. The first method that I will be talking about is autoclaving. This is a heat-related method. Autoclaving is very effective at killing organisms since it denatures proteins and destroys the cytoplasmic membrane. Autoclave works by raising the temperature higher than the water boiling point, which is 100 degrees Celsius, and pressure is applied to prevent the steam from escaping. As the steam enters the autoclave chamber, the temperature increases as well as the pressure until the set temperature and pressure are reached. Scientists have determined that the thermal dead point, which is the lowest temperature that kills the cells, is 121 degrees Celsius, and the thermal dead time is the time that takes to sterilize a liquid is 15 minutes. However, in solid substances, it requires extra time to sterilize because it takes more time for heat to penetrate in the center. Autoclaving is effective at killing endospores because the sterility indicators such as endospore test ampule is used to determine if the contents have been sterilized, since the ampule contains growth medium and a pH indicator, and when it's broken, it exposes the endospores to the medium, and if the endospore is still alive, the pH of the medium decreases, changing its color, and if there is no color change, it indicates its sterility. The second method that I will be talking about is incineration. Incineration works by oxidizing everything completely, and it only takes some seconds to sterilize at 1500 degrees Celsius. This method is used in microbiology labs to sterilize inoculating loops and inoculating needles with a Bunsen burner until they glow red, or an electric heating coil can also be used. This method is also used by healthcare workers by incinerating contaminated dressings, banks, and paper cups. Incineration is effective at killing endospores because it destroys organisms completely. Another example that our book mentioned is that epidemiologists incinerate animal carcasses that have been infected with highly infectious bacteria or viruses such as anthrax and bovine spongiform cephalitis. Ionizing radiation is the third method that I'll be talking about. Ionizing radiation destroys DNA depending on the wavelength of radiation. It can take seconds to hours of exposure. Cathode ray machines produce electron beams, which are very effective at killing microbes. However, they can't sterilize thick objects. These are usually used in dental and medical supplies, and they use it on gloves, syringes, and suturing material. Gamma rays penetrate farther than electron beams, and this method has been approved by the FDA by irradiating meats, fresh fruits, and vegetables. Since it kills the microbes, it also prevents food spoilage. X-rays have less energy than gamma rays and are not that suitable because they take a long time to kill the microbes. Ionizing radiation will be effective at killing endospores since it is used to sterilize medical and laboratory equipment, and they also help to preserve food as well. It's just that they just take a longer time to kill them, and it's harder to penetrate thicker objects. Thank you.